Culture, bacterial, blood, aerobic, with isolation and presumptive identification of isolates, includes anaerobic culture, if appropriate. Samples for bacterial blood culture are drawn by venipuncture and usually consist of a set of bottles, an aerobic and an anaerobic bottle. The test includes gram staining and subculturing to selective media for the detection of bacterial growth. There are several automated systems that detect the presence of bacteria using colorimetric, radiometric, or spectrophotometric means. Culture, bacterial, stool, aerobic, with isolation and preliminary examination, e.g., Kia, Lea, Salmonella, and Shigella species. This test may be called a stool culture, culture for Salmonella and Shigella, or routine culture when stool or rectal swab is the specimen. The testing method includes gram staining and subculturing to selective media for the detection of bacterial growth. Culture, bacterial, stool, aerobic, additional pathogens, isolation and presumptive identification of isolates, each plate. The test includes gram staining and subculturing to selective media for the detection of bacterial growth. Culture, bacterial, any other source except urine, blood, or stool, aerobic, with isolation and presumptive identification of isolates. Culture, bacterial, any source, except blood, anaerobic with isolation and presumptive identification of isolates. Culture, bacterial, with isolation and presumptive identification of isolates, any source except urine, blood, or stool, quantitative, aerobic. Quantitative, anaerobic. Common names for this test are numerous and may include routine culture, aerobic culture, or, using a body or source site, may be referred to as vaginal culture, cerebral spinal fluid culture, etc. Culture, bacterial, additional methods required for definitive identification, each isolate, anaerobic isolate. Aerobic isolate. Culture, presumptive, pathogenic organisms, screening only. With colony estimation from density chart. Culture, bacterial, quantitative colony count, urine. Culture, bacterial, with isolation and presumptive identification of each isolate, urine. Culture, fungi, mold or yeast, isolation, with presumptive identification of isolates, other source, except blood. Blood. Fungal culture, yeast culture, and mold culture are common names for this procedure. This test is to culture and isolate fungi, yeast or mold, with presumptive identification. Presumptive identification may include fungi, yeast or mold, present or a genus name with no species, e.g., Aspergillus. Culture, fungi, definitive identification, each organism, yeast. This test is commonly known as a fungal yeast identification. Various identification procedures, including growth patterns, and macroscopic and microscopic characteristics, are employed. Examples of fungal yeast pathogens that might require definitive identification include Histoplasma, Coccidioides, and Blastomyx. Culture, Mycoplasma, any source. Culture, Chlamydia, any source. The test method is by cell culture, fluorescent stain. The cell culture technique is to isolate for chlamydia. Culture, tubercle or other acid-fast bacilli, e.g., TB, AFB, Mycobacteria, any source with isolation and presumptive identification of isolates. Common names include AFB culture, TB culture, Mycobacterium culture, and acid-fast culture. Collection methods are source-dependent. The methodology is by culture for the isolation and presumptive identification of Mycobacterium. An acid-fast smear should be done at the time the specimen is cultured. Media for isolation should include both solid and liquid types. Culture, mycobacterial, definitive identification, each isolate. Methodology is traditional biochemical tests for identification of Mycobacterium. Culture, typing, immunofluorescent method, each antiserum. Specific antisera are combined with a fluorescent dye and used to stain slides of organisms. Stained slides are scanned with a fluorescent microscope to look for fluorescing organisms. Typing of organisms by immunofluorescent technique is usually to determine whether an organism is of a more pathogenic strain, to determine a treatment, or for epidemiological purposes. Culture, typing, gas-liquid chromatography, GLC, or high-pressure liquid chromatography, HPLC, method. The methodology is gas-liquid chromatography, GLC, or high-pressure liquid chromatography, HPLC, to analyze byproducts of rapidly growing organisms. Culture, typing, immunologic method, other than immunofluorescence, e.g., agglutination grouping, per antiserum. This test is used for more specifically identifying cultured specimens using an immunologic method other than immunofluorescence. For example, agglutination technique may be used to more specifically identify Salmonella, usually to a group level, since there are more than 2,000 seravar of Salmonella. Culture, typing, identification by nucleic acid, DNA or RNA, probe, direct probe technique, per culture or isolate, each organism probed. Amplified probe technique, per culture or isolate, each organism probed. Culture, typing, identification by pulse field gel typing. This test may also be known by the acronym PFGE, Pulsed Field Gel Electrophoresis, SHEF, Contour Clamped Homogeneous Electric Field, or a combination of the two. A bacterial culture and nucleic acid isolation precedes the test, and cells are harvested by centrifugation and washing. The PFGE method resolves and separates very large DNA molecules for cloning and direct visualization of small chromosomes, among other genetic analysis. Culture, typing, identification by nucleic acid sequencing method, each isolate, e.g., sequencing of the 16S rRNA gene. Culture, typing, other methods.